Hi, everyone. Welcome, welcome. All right. Very good. If you would, go ahead and turn your video on, but keep your audio off. Keep yourself muted, but if you wouldn't mind, turn your video camera on. Thank you. All right, very good. Well, welcome to a Zoom lecture. I apologize. I apologize that I'm not with you today. I've been battling a bit of a cold or something, some kind of a respiratory infection. I'm all hopped up now on on Mucinex, so hopefully I won't do too much coughing or sneezing while we're together today. But yeah, out of an abundance of caution, I decided to stay home today, and that's why we're meeting over Zoom like this, just to be safe. We're living in such strange times. As I said in my message, I did get a negative COVID test back, but remember, sometimes when you get retested, it can become positive and all that. I don't think I have COVID, but just to be safe, I'm staying home and keeping my germs to myself today. So what we're going to be doing today is finishing up our discussion of cellular respiration, oxidative phosphorylation, and fermentation. So the second half of this lecture topic, essentially. Oh, Emma, thank you for letting me know. Yeah, I appreciate that. Sometimes we have to do some crazy things in order to get everybody together on Zoom. So I appreciate it when you help each other out like that. It's great. Just a couple of housekeeping notes before we start. When I run Zoom lectures, I do like to have video on, audio off on your end. So I make use of the chat quite extensively. I find that everything streams a little better if we have audio off. So if you have a question to come up while I'm talking, go ahead and type it right into the chat. Just type question and then whatever it is, and I'll address it. I'll also stop periodically like I do when we're together in the classroom, and I'll ask if anybody has any questions. Okay, so we'll make use of the chat more than anything. If you need to step away, if you need to, I don't know, take a phone call, or if somebody walks into your room and you need to go away from your camera, if you need to turn your video off for a minute or two, that's fine. Go right ahead. I do like to see you, though. It just makes me feel better if I'm actually lecturing to faces as opposed to black screens or just names. It's a little strange when you lecture over Zoom, if you've never done it. The whole thing is a little strange. But again, I'm glad we have it available right now. Another housekeeping note, this Thursday, we don't meet for lecture. Thursday is the 11th and it's Veterans Day. So we will not be meeting for class. That'll be a day off for you, in other words. And then looking ahead to next week, on Tuesday, we will meet. We're not going to have any new material introduced on Tuesday. We're going to have a discussion. I'm just not sure exactly what we're going to be doing yet. Originally, I had hoped to do some kind of a lab exercise with you related to aerobic cellular respiration. That has not come together in time. So I'm still putting together what we're going to be talking about that day, but it will essentially be an informal type of a discussion, as you could even think of it maybe as a dry lab. We won't be doing an experiment together. but we will have some kind of a discussion related to what we've been talking about in class. Now, on Thursday next week is your next exam. And it's going to cover all of the lecture material since your last exam. So it will start with the material about energy and enzymes and cellular respiration, oxidative phosphorylation, which we'll talk about today, and fermentation, which we'll talk about today. So everything since the last exam, starting with that energy lecture, through what we discussed today will be on the exam. And as always, I always recommend that you get started studying as soon as you have all the material available to you. This material about cellular respiration, oxidative phosphorylation, and fermentation is very difficult, as you know, and it can take a while to get it under your belt. So, so use the time you have. You'll have all the information you need after class today. And then the exam won't be for another, 10 days. So use that time to make sure that you've got the information mastered. All right. Does anybody have any questions about the first half of this lecture, which was cellular respiration, essentially? Does anybody have any questions about what we talked through the last time we were together? We primarily covered glycolysis, pyruvate processing, and the Krebs cycle. We're going to start today with the fourth set of reactions in cellular respiration, which is electron transport. That's where we'll be starting today. But if anybody has any questions about anything we talked about last time, we can certainly begin with those. Okay. All right. Well, before I open up the slides for us, I'll have you do one more thing for me, for my records. And if you would, just type your first name or your nickname for me into chat. Just type in your first name for me. Great, thank you. That's just a way for me to have a written record of who attended today, because Zoom will keep that for me in a document. Zoom will keep that chat for me in a document. Thank you, that's wonderful. Very good. All right, so I'm going to open up the slides now. I'm going to share my screen with you and open up the slides. So what you should be seeing now, hopefully, is a slide that says electron transport at the top. Hopefully that's what you're seeing on your screen. Electron transport. So remember, just to review, when we talk about cellular respiration, there are 4 sets of reactions involved. There's glycolysis, pyruvate processing, Krebs cycle, and electron transport. At the end of electron transport, we're done with cellular respiration. We haven't yet made all the ATP because that comes in the next process, which is called oxidative phosphorylation. So cellular respiration is often discussed as part of a process that involves oxidative phosphorylation. Okay. Cellular respiration is this series of 4 reactions, beginning with glycolysis and ending with electron transport. Remember, what goes into glycolysis is glucose. And as we talked through those reactions, we talked about what goes in, what comes out, whether or not any ATP is made. As the glucose is broken apart bit by bit by bit, what molecules are being created. at each step of the way. For example, in glycolysis, glucose goes in, glucose enters, and pyruvate comes out. And along the way, we said a little bit of ATP gets made. We also load these molecules called electron carriers or electron acceptors. We're removing electrons from glucose. Glucose is getting oxidized. It's losing electrons because we're breaking it apart. We're breaking covalent bonds. And in each covalent bond, there's a pair of electrons. So glucose loses the electrons, and that means something else has to gain them. And that something else in glycolysis is this molecule called NAD+. It's going to gain the electrons. It's going to get reduced and become NADH. So a little bit of ATP gets made in glycolysis. We said no ATP gets made in pyruvate processing. We said a little bit of ATP gets made during Krebs cycle. And remember, the ATP that gets made during glycolysis and during Krebs cycle is made using that process called substrate level phosphorylation. We're going to make a molecule of ATP sort of by piggybacking on top of some enzymatic reaction. We're going to phosphorylate an ADP by adding a phosphate group to it and rebuilding ATP. That's substrate level phosphorylation. That's a really bad way to make ATP for survival because it's very inefficient. Remember, in the Krebs cycle, we said there's a second electron transport molecule or electron carrier or electron acceptor molecule. It's called FAD. And it becomes FADH2 after it accepts electrons, after it gets reduced. So electron transport is what happens towards the end of cellular respiration. By the time we go into electron transport, glucose has been broken all the way apart into CO2, into carbon dioxide. There are no covalent bonds left to break in glucose. But we now have all of these electron carrier molecules, these electron acceptor molecules that are fully loaded with electrons. That's NADH and FADH2. And we haven't made any ATP yet. So the last part of electron transport is going to involve those electron carrier molecules, these two molecules here, becoming oxidized. In other words, they're going to give up the electrons that they're carrying into a structure called the electron transport chain, or the ETC. This is what we mean when we talk about electron transport. All the ETC is a group of proteins, specialized proteins that are embedded in the inner mitochondrial membrane. They're embedded together in clusters. We call them complexes. What makes these proteins special is that they have different electronegativities. So they are more or less attracted, if you will, to electrons. Remember, things that have a higher electronegativity are able to pull electrons more closely to themselves. So we're going to pass the electrons from protein to protein to protein in this electron transport chain. And just like the rest of cellular respiration, one thing gets oxidized, something else gets reduced. Then the next thing gets oxidized, and then something else gets reduced. We're walking the electrons down this series of proteins. Now, my best analogy for an electron transport chain is a staircase. If you think about the electron transport chain as a staircase, I think it's helpful. The electrons are going to go down the chain. That's the language that we use. They're going to be passed down the chain, just the same way something would go down a series of steps in a staircase. And along the way, those electrons are going to have their energy harvested from them. This is the weirdest concept, I think, in all of cellular respiration. We've talked how electrons are the conduits of energy. If you want to watch how energy is moving around in a cell, all you have to do is watch what's happening with the electrons. Well, in the chain, as the electrons get passed down, their energy is sort of taken from them. It's removed from them. They're still electrons. They're still electrons, the same little particle that we've been talking about all semester. At the end of the chain, something is going to have to receive them. Something at the end of the chain is going to have to accept these electrons. Because remember, every time something gets oxidized, something else has to get reduced. At the end of the electron chain, at least in humans and other animals that breathe oxygen, the final electron acceptor molecule is oxygen gas. So these electrons get stepped down the chain. And at the bottom, they'll be accepted, they'll be received by oxygen. That's going to create water, and we'll be done essentially with the electrons that we harvested from glucose. But we've taken their energy out. We've essentially harvested their energy from them, and we're going to use that energy to make ATP. We're going to take the free energy out of the electrons, and we're going to build ATP with it. All right, that's what happens during oxidative phosphorylation. But this, remember, this is the final part of respiration, the final part of cellular respiration, this process where these electrons travel down the electron transport chain and get received by by oxygen. Now, the reason I like the analogy of the staircase is because most of us, maybe not all of us, because I'm a little older than most of you, but most of us know what a slinky is, right? You may have had a slinky when you were a little kid. A slinky is just a little metal spring that kids play with. It's a toy. And one of the things that they used to advertise that you could do with your slinky is you could put it at the top of a staircase and you could set it in motion and it would sort of walk down the stairs. And of course, if you've ever taken a physics class, you know that that spring, that slinky, has potential energy. The staircase, in fact, has potential energy. And by putting that slinky in motion, that spring in motion, we can take advantage. We can release that energy and it will carry that slinky all the way down the stairs. And at the bottom of the stairs, the slinky will stop. So this spring and this staircase make a nice analogy for the electron transport chain, because at the top of the chain, at the first high stair, the electrons have a lot of energy associated with them. But by the time they travel from step to step to step down the staircase, some of that energy is being released and it's being harvested for use somewhere else. So by the bottom of the staircase, just like the slinky will stop, at the bottom of the staircase, the electrons have given up their potential energy. And they will be received by oxygen so that water can be created and the oxidation reduction reaction is complete. So I do like that analogy, and I tend to use the staircase analogy a lot when I talk about the ETC. What's important to remember, though, is that this cellular staircase is made out of protein. And the reason it works is because these proteins have specialized structure that allows them to differ in their electronegativity and pass the electrons from one to another, to another, to another, like the slinky going down the stairs. This is just more oxidation reduction chemistry. The NADH and the FADH2, those are the two electron acceptor molecules that got loaded up with electrons. They're going to lose electrons at the top of the staircase, at the top of the ETC. They're going to become oxidized. They're going to donate, if you will, those electrons into the electron transport chain. So they were reduced, those little molecules, they were reduced when they gained electrons. Now they're going to get oxidized. Now they're going to donate the electrons into the ETC. The next thing that's going to happen is, again, the electrons get moved down the staircase. One protein, the protein that received them at the top of the staircase, that protein just got reduced. It gained electrons. So it's going to donate the electrons to the next protein. And the first one gets oxidized, the second one gets reduced. Then the second one will get oxidized and give the electrons to the next protein. And then that one gets reduced. And we just keep going, oxidation reduction through these complexes of specialized proteins until we've taken all of the energy out of those electrons. Thanks, Josie. I see your message. Thanks for that. Thanks for helping her out. All right, let's go back into the slides now. So what's happening to the electrons in the electron transport chain is they're being passed from protein to protein to protein through oxidation reduction reactions. And their energy is being taken from them. It's being harvested from them. At the bottom of the chain, there's oxygen. Oxygen is going to get reduced. There will be one final oxidation and then one final reduction where oxygen receives all of those electrons that were released from glucose. And remember, the oxygen plus the electrons plus a little bit of hydrogen is going to produce water. Now, what about this energy? What about the energy that got harvested as they went down that staircase, that electron transport chain? This energy, this energy is going to be used. It's going to be used, and it's going to be used to pump protons. And remember, that's just another name for a hydrogen ion. We're going to pump protons across the inner mitochondrial membrane into the intermembrane space. Now let's orient ourselves for a minute. This pump, remember what a pump is in a cell. It's a protein that can maintain a gradient. It can build and maintain a gradient, and it requires energy in order to function. So this pump is going to move hydrogen ions from the matrix across the inner mitochondrial membrane into the intermembrane space. Remember, there's an outer mitochondrial membrane and there's an inner mitochondrial membrane, and there's a space between them. That's the intermembrane space. There's also a space, a cavity, if you will, within the inner mitochondrial membrane. That's where the matrix is. So we're going from the matrix across a membrane into the intermembrane space. And this pump is going to build up a nice big gradient of hydrogen ions. Remember, a gradient, an ion gradient, is a place where you can store energy just for the very short term. So the energy that came from the electrons in the electron transport chain will be very short term stored in a hydrogen ion gradient. Now we still haven't used it. We've moved it from one place to another, this energy, but we still haven't used it. This is where we use it. There's a protein, it happens to be an enzyme, sitting in the inner mitochondrial membrane, right near the electron transport chain, called ATP synthase. This is an amazing protein. This is an amazing enzyme, and we'll talk about it structurally in just a minute. What's going to happen is the hydrogen ion gradient that gets built by this pump and with the energy that was harvested, this hydrogen ion gradient is going to be allowed to resolve once it's in place. It is going to physically flow through this enzyme called ATP synthase. This enzyme is sitting in the membrane. It's embedded in the membrane, and it's one of those transmembrane proteins. It passes all the way through the membrane from one side to the other. And it has a tunnel in the middle of it, just like a channel protein. But it also can do something amazing. While the hydrogen ions are flowing through this protein, through the tunnel that this protein creates, it's going to be making ATP. So take a look at the drawing on this slide. This represents the inner mitochondrial membrane. And just like all membranes that we've seen, you can see the heads on one side, the heads on the other side, and all the tails in between. So to orient us, there is an outer mitochondrial membrane somewhere. And in this drawing, it would be over here somewhere. We just can't see it. There is a space between the two membranes called the intermembrane space. So that's what this is right here. Now, if we go inside the inner mitochondrial membrane, there's another space called the matrix. Okay, so the matrix would be over here. So there is a collection of very specialized proteins embedded in the membrane, the inner mitochondrial membrane, called the electron transport chain. And that's what this is. That's what you're looking at here. What's labeled number 1 is actually a group of proteins, a complex of proteins. What's labeled 2 is just another group of proteins. And then there's 3, and then there's 4. So there are 4 sets of proteins, 4 complexes of proteins in the electron transport chain. And they're quite specialized because they're going to be very good, very effective at transporting electrons because of their differing electronegativities. Now, there are 2 other things to note here. There's something called Q here, and there's something called site C here. We'll talk about those in just a minute. But notice what's happening right up at complex 1. Here's an NADH. So this is that electron acceptor molecule that got loaded up with electrons during the first 3 reactions of cellular respiration. The arrow is showing us that it's giving up its electrons. It's donating its electrons to this first complex of protein. And this complex is going to pass those electrons from one protein to the other. Oxidation, reduction, oxidation, reduction. Sorry, I have a very sensitive mouse in my hands here at home. Energy is going to be harvested from those electrons as they're passed from one protein to the other in this complex. The energy that's gathered from those electrons is going to be used to build a gradient. And you'll notice something is happening here. There's an arrow drawn right through the complex from the matrix side over to the intermembrane space side. Hydrogen ions are being transported. That's right, the pump that moves hydrogen ions is actually part of the complex of proteins. So as that complex sends the electrons from protein to protein and harvests their energy, it immediately uses their energy to pump hydrogen ions into the intermembrane space. Now, once the electrons have gone all the way through complex 1, they're going to be transported to complex 2, and then to complex 3, and then to complex 4. Same thing is happening through all these complexes. The electrons are being passed from one protein to another. Their energy is being harvested and used to pump hydrogen ions. Notice that the FADH2 carrier didn't donate its electrons to complex 1 like NADH did. Instead, it starts by donating its electrons to complex 2. Don't worry about that detail too much. Just understand that these two molecules, NADH and FADH2, are going to come to the electron transport chain, and they're going to donate their electrons into it. That's the most important thing to understand. Those electrons then get passed through these complexes. At the very end, when the electrons come out of complex 4, they're going to be received by oxygen. The electrons plus the oxygen plus a little bit of hydrogen is going to create water. So we've finished with the electrons. We took them out of NADH and FADH2. These two molecules became oxidized. We had a series of oxidation reduction reactions. And then the final reaction was a reduction. Oxygen got reduced. It received all the electrons that come out of the chain, and we create water. And along the way, we've pumped a lot of hydrogen out of the matrix and into the intermembrane space. Now, what about these two strange little molecules? This one's labeled Q, this one's labeled site C. These are actually helper molecules. These molecules are going to assist in taking the electrons from one complex to another complex. So the electrons get passed down the proteins in complex 1. Then they're going to get handed to Q, and Q is going to bring them to the next complex. It happens to be complex 3. Don't worry about that detail too much. Just know that Q is kind of a transporter. Q is going to help get the electrons from one complex to another complex. Cytochrome C does the same thing. Cytochrome C and Q are molecules that just help. They help get electrons where they need to go as they travel down this electron transport chain. Now, interestingly enough, that Q molecule goes by a couple of different names. The chemical itself, the molecule itself, is called ubiquinone. And I'll show you that name on the next slide. But ubiquinone is also known as coenzyme Q. And we often shorthand that second name as Q. So ubiquinone, coenzyme Q, and Q are all the same molecule. If you go into a pharmacy, in fact, the next time you go to a CVS or a Rite Aid or wherever you happen to go to the nutritional supplements wall, because it's usually a wall, right? And look, and you will find a bottle labeled coenzyme Q. There are people who believe that if they take coenzyme Q, they will help their cells make more ATP. Now, I've mentioned supplements before in this class, I think. I can never remember who I've said what to in my courses, but in reality, the human body is very careful about how much of certain molecules are allowed at any one time. Because a lot of the way that the body maintains homeostasis is through feedback. We talked about feedback with the glycolysis enzymes and the pyruvate processing enzyme. We talked about how cells can control how much enzyme activity goes on because they don't want to make any product that they don't need. The other thing cells don't do is they don't store ATP. So there are some people who think they can sort of trick cells to make more ATP for them so they'll have more energy. But in reality, that doesn't really happen. Now, having said that, any person who is deficient in ubiquinone will benefit from taking ubiquinone, right? If you don't have enough ubiquinone because something's wrong, then taking a ubiquinone supplement would be helpful for you because all you're doing is returning it to its regular normal level. There are some people who have certain heart conditions, certain cardiac diseases, and their coenzyme Q levels do tend to be low. So for those people, it makes sense to take a coenzyme Q supplement. That supplement is just going to bring their ubiquinone levels back to normal. But no, you can't trick your body into making extra ATP for you. It's just not going to work. So I always warn people about the magic of supplements. Supplements are great if you are deficient in something. But beyond that, they're really just expensive frauds in a lot of cases. Yeah. All right. That's my supplement talk for today. So the electron transport chain is embedded in the inner mitochondrial membrane. The proteins are grouped into what we call complexes. There are 4 of them. And these are unique proteins. They have these strange little additional molecules bound to them so that they can have this range of electronegativity. You don't need to know these specific names. I'm just showing you that there are molecules, for example, called flavins. There are molecules called iron sulfur complexes. There's molecules like heme that are added on to these proteins so that we can set up a chain of proteins that have differing negativities. So you don't need to be able to tell me these names, but understand that the proteins in the ETC are kind of unusual. They're not like a lot of the other proteins that you see in cells. They're designed basically for electron transport, for receiving and then donating electrons. Whoops, that molecule that helps pass a protein from one to another, pass electrons from one protein complex to another, that's called ubiquinone. Ubiquinone is not a protein. Ubiquinone belongs to a class of chemicals called quinones. And the reason it got called ubiquinone is because scientists realized that it was quite ubiquitous. It was found everywhere you looked. In other words, in any eukaryotic cell, remember eukaryotes have organelles, in any eukaryotic cell that has mitochondria, you're going to find ubiquinone. So they wanted to give it a name that meant ubiquitous quinone, and that's where the name came from. But it's also known as coenzyme Q or just Q. All 3 of those names are for the same molecule. Q is a really interesting molecule because it is a quinone. They have strange, I think, strange chemical properties. They are lipid soluble. That makes sense. In other words, they're non-polar molecules. And that makes sense because this molecule has to essentially live in the tail section of that membrane. So it can receive electrons from one complex and then donate them to the next complex, and it physically moves through the membrane with ease because of its chemistry, because it's non-polar. It gets along well with the tail section. Here's where I think, again, it's kind of strange. It can also act as a pump. But hold on, we'll talk about that a little more in just a second. Now, in contrast, the complexes of proteins that make up the electron transport chain, they can't move. They're embedded. They're embedded in the membrane and they are sort of anchored in place. Now, if you think about it, that's important. That's important. We talked about our pool analogy, remember, when we talked about membranes? We said that phospholipids can actually move. They can actually float through a membrane. And the proteins that are embedded in a membrane, they kind of move around too, just like pool toys do on the surface of the pool. Lots of proteins, if you look at a spot on a membrane, a lot of proteins won't be there. If you come back 2 minutes later, they will have floated off somewhere else in the membrane. But there are some proteins that are anchored in place. And why this is important is because we need complex 1 to be next to complex 2, which is next to complex 3, which is next to complex 4. We need those 4 complexes to stay in order, and we need them to stay together. So it makes sense that they're physically anchored in the membrane. Now, it's important to know that there isn't one electron transport chain in your mitochondria. There are many of them. There are many, many electron transport chains throughout your inner mitochondrial membrane. And that's so as many electrons as possible can get processed through that chain as quickly as possible. The proteins inside the electron transport chain differ in electronegativity. So that's why they're able to receive and then donate, and then receive and then donate. That's oxidation and reduction, with Q acting as the sort of the messenger, the go-between. Q is helping pass the electrons from one complex to another throughout the chain. This is a diagram out of a textbook. You've seen something similar to this diagram. You'll notice the coloring scheme. It should be familiar to you by now. In this particular textbook, the matrix gets this orangey red color. The inner mitochondrial membrane is shown in blue, and then the intermembrane space is shown in blue as well. So that's where we are in blue intermembrane space. The membrane happens to be the inner mitochondrial membrane, and this happens to be the matrix. So they've drawn the electron transport chain in here for us. This big glob that's colored green represents a group of proteins, and those proteins together are known as complex 1. Here's complex 2, here's complex 3, and complex 4. Electrons are going to come into the electron transport chain. They're going to be passed from protein to protein to protein, all the way through the complex. And when they come out, they'll be combined with oxygen to make water. Along the way, the energy is being harvested. And the energy that's harvested is used to immediately pump hydrogen ions from the matrix side to the intermembrane space side. That's what they're showing us here. So we're building up a gradient. Lots of hydrogen ion over here, not so much over here. This is achieved by pump proteins that are part of these complexes. Also happen to be part of this molecule right here called Q. Here's Q taking some electrons and transporting them somewhere else, and also helping to pump hydrogen ions along the way. Remember, there is another molecule. This molecule is called cytochrome C. And it's also involved in transporting electrons from complex to complex, although cytochrome C is generally only involved in the later part of electron transport. All right. Both Q and cytochrome C are helping move those electrons from complex to complex. The energy is used, or it's taken from electrons and used to pump hydrogen ions. Q is, it says Q is the pump. Q is one of the pumps. The complexes themselves also contain pumps. And hydrogen ions build up in this intermembrane space. Lots of hydrogen ions here, not so many in the matrix. So now we've built a gradient, and ion gradients store energy for very short periods of time. We're going to very quickly release that energy from that gradient and use it to make ATP. So all those hydrogen ions that have been moved over into the intermembrane space, we're going to turn the pumps off and we're going to allow those hydrogen ions to flow back into the matrix through a channel in the membrane, through a tunnel in the membrane. And that tunnel is in this protein called ATP synthase. The energy in that ion gradient, in other words, is being transferred to the ATP synthase as the hydrogen ions pass through. And it's ATP synthase that will build ATP. This process, the process of using a hydrogen ion gradient to build ATP, is what's called oxidative phosphorylation. Remember, this word has nothing to do with oxygen. This has nothing to do with oxygen. This has to do with oxidation reduction chemistry. Oxidative or oxidation reduction chemistry, phosphorylation. That word just means we're going to put a phosphate group onto ADP to build ATP. All right. You can see where I had that lecture split. What we really want to do now is take a look at ATP synthase, because this is probably one of, if not the most interesting proteins in the human body. ATP synthase is big. It's a protein, so of course it's big, but it's a particularly big protein. If you remember when we talked about pyruvate processing and we talked about the enzyme that does that work called pyruvate dehydrogenase, or PDH, we said that enzyme is called an enzyme complex because it's massive. It's gigantic. It's even bigger than most other enzymes. ATP synthase is another enzyme complex. It's huge, relatively speaking, and it's actually divided into a couple of different parts. There's a part that's embedded in the membrane near the electron transport chain. And then there's a part that hangs off of it, sort of hangs off of it, literally. When hydrogen ions flow through the part that's in the membrane, because remember that part forms a tunnel in the membrane. When it flows through, those ions flow through the tunnel in the membrane, that is going to cause the part that's hanging down to physically spin. It's going to physically spin. It's fascinating if you're a geek like I am. They have hydrogen ions flowing down their gradient, flowing through the channel portion of ATP synthase, is going to cause the second half of ATP synthase to move. And it's that kinetic energy, that motion, that is going to phosphorylate ADP and make ATP. The other thing that I find fascinating about ATP synthase is that that enzyme is found in every single cell on our planet. Not all organisms perform aerobic cellular respiration. We talked about that. We said some organisms do cellular respiration, but they don't use oxygen as the final electron acceptor. They use some other molecule. There are a lot of, for example, anaerobic bacteria that use things like nitrate and sulfate and carbonate. You don't need to know those names, but they use other molecules at the bottom of the electron transport chain to accept all the electrons that come down. They don't need oxygen to do respiration like we do. They need a different kind of molecule. What's fascinating is that we can be so different. You and I can be aerobic, eukaryotes. They can be anaerobic, prokaryotes, and yet both of us have ATP synthase. And we use our ATP synthase to build ATP. The other interesting thing is that prokaryotes don't have mitochondria, do they? Prokaryotes don't have any organelles. Bacteria don't have organelles. Archaea don't have organelles. So where the heck is their ATP synthase? The answer is it's in their cell membrane. It sits in their cell membrane, their plasma membrane. They still use electron transport. They still have an electron transport chain. They're still going to build a hydrogen ion gradient. They don't do it in mitochondria, though. They do it across their cell membrane. And they allow the hydrogen ions to flow back through the ATP synthase enzyme, and that causes ATP to be produced. So that ATP synthase enzyme, it's one of the few molecules that you can find in every single organism. It's kind of fascinating, I think, anyway. So ATP synthase has two parts. It's got one part that's often called the knob. That part is technically called the F1 unit. And that's the part of the enzyme that's bound to the membrane. It's literally sitting in the membrane. Then it's also got this other part called the F0 or F0. That's how we say that, the F0 unit. The two units are connected to each other by basically a stalk. Hydrogen ions or protons are going to be allowed to flow through that F0 unit and cause the stalk to spin. Sorry, that's written incorrectly. The hydrogen ions are going to flow through the F1 unit. That's a typo. The hydrogen ions are going to flow through the F1 unit, the part that's in the membrane. Just remember that. They flow through the part that's in the membrane, and they cause the part that's hanging below the membrane to spin. As it spins, it changes shape. Remember, this is how the cell controls proteins, by changing their shape. And we've talked about how you can stick something onto a protein and make it change shape. Well, in this case, it's the mechanical motion of that thing spinning that's going to cause it to change shape. That's going to allow a phosphate group to be added to ADP and will make ATP. This is oxidative phosphorylation. And this is extremely efficient. We're going to get a lot of ATP for a little bit of glucose. Now, take a look at the drawing down here on the bottom. This represents ATP synthase. So here's the F0 unit, and it's embedded in the membrane. Here's the F1 unit hanging below. And notice it's hanging down into the matrix. There's a little stalk that joins those two units together. So when you look at this strange green thing, this is the actual structure of this molecule. This molecule has been studied so much that we know exactly what it looks like. We know exactly how it's shaped, how it folds. Up here, it's really hard to see, but up here is an image of some ATP synthase molecules embedded in a membrane. And this was taken with an electron microscope. Now, it's very hard to orient yourself here, but what we often refer to these as is lollipop and stick structures. So this would be the lollipop, and this would be the stick. So you can see right here, this represents a lollipop unit. There's another one over here. Don't worry if you can't really make them out. They're hard to see. Here's another lollipop right here. They're hard to see, but understand that we know exactly how this protein is shaped because we've been studying it so long. What's going to happen after the hydrogen ion gradient is built up in the intermembrane space? We're going to allow it to resolve. We're essentially going to turn the pumps off. All those hydrogen ions are going to do what gradients do. They're going to flow down their concentration gradient. So from the side with a high concentration towards the side with a low concentration. And they're going to flow through this protein because this protein forms a channel in the membrane. It forms a tunnel through the membrane. So they'll go from this side through the ATP synthase F0 unit into the matrix. And that motion is going to cause this to change shape and to spin. This thing literally physically turns. And the energy, that mechanical energy of this thing spinning or turning, is going to take a phosphate group, add it to ADP, and make ATP. Absolutely bizarre. I know of no other enzyme in your body that works like that. I know of no other enzyme that spins around like that. But that's how this enzyme works. And because so many electrons get harvested from one glucose, and so much energy can be harvested from those electrons, we can make lots of individual ATP molecules from one glucose. Remember, I said, depending on the source that you look at, you'll read anywhere from 26 to 30 and sometimes 32 molecules of ATP for each glucose. Don't worry about the actual numbers. I'm not going to test you on that. But understand that it's a very efficient process. You get multiple ATP molecules made for each glucose that goes through cellular respiration. That's very different from substrate level phosphorylation, which was the one where we got one ATP every time the enzyme did its reaction. Some enzyme is doing a chemical reaction, and we're getting one ATP made in the process. That's inefficient. Oxidative phosphorylation is very efficient, and it's the way we make most of our ATP. You can imagine that if you were born with a defect in the gene that builds ATP synthase, you would not survive. In fact, you probably wouldn't get very far along at all through embryonic development. There are people, though, who are alive, who are walking around, who aren't missing ATP synthase, but they are missing pieces and parts of this system in their mitochondria. There are people that have deficiencies in enzymes, for example, enzymes involved in glycolysis or involved in parts of the Krebs cycle. And these people cannot make normal levels of ATP because of it. These people who have these mitochondrial diseases, they often inherit them from mother and father. Luckily, they're very rare. And while some of them are survivable, you can imagine that it greatly impacts their quality of life. If your mitochondria can't do this fundamental reaction normally and make ATP normally, your cells are going to suffer for it. And people who have mitochondrial problems often suffer with an inability to do even some of the most basic things that you and I do every day because they simply do not have the ATP to do it. Remember, one of the systems in your body that consumes a whole lot of ATP is the musculoskeletal system. And people who inherit abnormalities in their mitochondria are often bedridden. They simply don't have enough ATP to do even the most basic work in their cells every day. They also don't tend to live long lives. They tend to die prematurely because of that. So luckily, mitochondrial defects are quite rare, but they do occur in some people. All right. If you remember, we looked at this diagram in part the last time we were together. It showed us a little bit of, we looked at where glucose went through glycolysis out in the cytoplasm and became pyruvate. And along the way, we loaded up our electron carrier molecule. We made a little ATP. We said the pyruvate then moves into the mitochondria. That's what this represents over here. It goes through pyruvate processing and we get acetyl-CoA and we get carbon dioxide. And we load up some more NADH. Then the acetyl-CoA goes into the Krebs cycle. Remember, it joins with a molecule called oxaloacetate. And this cycle just goes round and round. Along the way, we load up more NADH as well as a molecule called FADH2. We make a little ATP along the way. And by the end, all we're left with is carbon dioxide. We started with glucose and we broke it all the way apart. We oxidized it all the way down to carbon dioxide. All the electrons got accepted temporarily by these molecules. Well, now we can add the rest of the picture in. Now, once the glucose has been fully oxidized all the way down to carbon dioxide by the end of the Krebs cycle, these electron carrier molecules are going to go to the electron transport chain embedded in the inner mitochondrial membrane. They're going to become oxidized. They're going to donate electrons into the chain, and the proteins inside the chain will be reduced. And then those proteins will pass the electrons to the next protein, and then the next, and then the next. Oxidation, reduction, and then oxidation, and then reduction, and so on down the chain. This little orange circle that you can just barely see, that represents ubiquinone, helping the electrons move from complex to complex. We can also see cytochrome C in green, moving electrons. Along the way, as the electrons are passed from protein to protein in the electron transport chain, their energy is being stolen from them. It's being harvested from them and used to pump protons from the matrix into the intermembrane space, building up a hydrogen ion gradient. At the end of the electron transport chain, oxygen is going to receive the electrons and we're going to get water made. This hydrogen ion gradient that we have built with the energy from all these electrons is going to be allowed to resolve itself by flowing down its concentration gradient through ATP synthase, which is right here. As the hydrogen ions flow through the tunnel portion in the membrane, they're going to cause the bottom half, the half that's hanging down below, to spin. And that spinning motion is going to cause ADP to become ATP through oxidative phosphorylation. So you can see in this diagram, they say 26 ATP will be made here, coupled with the other ATP, we're going to get a total of 30. Again, don't worry about that number, because different source materials you read will give you different numbers, unfortunately. But just know you get quite a good return for each glucose that passes down this long, long series of reactions. Now, a couple things to note. Remember, we said not every organism on this planet is an aerobe. Not every organism on this planet breathes oxygen and uses the oxygen at the end of their electron transport chain. You and I do aerobic cellular respiration. We use oxygen as our final electron acceptor at the bottom of the electron transport chain. But there are lots of organisms, especially bacteria and archaea, not all of them, but some of them, who do anaerobic cellular respiration. They're going to do the same set of reactions: glycolysis, pyruvate processing, Krebs cycle. Then they're going to send the electrons down the electron transport chain. But at the bottom of their electron transport chain, it's not oxygen, it's another molecule. It's things like nitrate or sulfate. There are others as well. They're doing the same thing. They're just using a different molecule at the very end to receive all of the electrons that were released from glucose. They still have an electron transport chain. They still have ATP synthase. They're just doing it a little bit differently than you and I are doing it. Now, the last thing we'll talk about today before I let you go is fermentation. This is one of my favorite subjects because one of the courses that I teach here at Keene State and at other colleges in the area, I teach microbiology. And a lot of fermentation, at least on a commercial level, is done with microbes. You are probably familiar with that, even if you didn't realize it, because a lot of products that you and I buy at the grocery store are produced through fermentation at commercial facilities that use microbes to make products that you and I like. Things like ethanol in our beer or our wine, things like sauerkraut and soy sauce and tofu. All of those things are produced through microbial fermentation. So it's a really important part of metabolism. In fact, you and I are also capable of a type of fermentation. We just don't use it each and every day. So let's think about what happens to you and I when we don't have oxygen available to us. Remember, we said oxygen is our final electron acceptor molecule. And if we stop breathing, we have a very short amount of time to survive because we need oxygen at the bottom of our electron transport chain, or else cellular respiration cannot occur. So what happens when we don't have any oxygen? Now, certainly, if you stop breathing, let's say you have a heart attack, you stop breathing, you fall to the ground, you're unconscious. That, of course, is a medical emergency. And the people who come to assist you the medical people who come to assist you are going to deal first with getting oxygen back into your system. That is perhaps even more important than getting your heart pumping normally again, because you are on a very limited clock with your oxygen, 4 to 6 minutes, depending on how old you are or what activity you were doing when you had your heart attack. But within 4 to 6 minutes, your cells are going to start to die. And your brain cells and your heart muscle cells are going to die and not be replaceable. So very, very dangerous situation if you stop breathing. But there are other times for us humans when it's not that we're not breathing, it's that we're not able to get enough oxygen for the amount of work we're doing. And one of the best examples of this is when we're exercising very vigorously. I love to use the example of the weekend warrior kind of athlete. You know the kind. The weekend warrior athlete is the person that, works all day during the week, and then on a Saturday might put on their running shoes and go for a 10-mile run. The person who's not really in shape, but maybe pushes themselves to do a high level of athletic performance. And of course, when you go out and you start running and you're not particularly in shape, what's the first thing that kind of happens to your breathing as you start to run? If you're not really in shape and you go out and you start running, what's going to happen with your breathing? Type it into chat. How's your breathing going to change as you start your 10-mile run on Saturday afternoon and you're a little bit out of shape? You're going to be breathing normally? You're going to have the same respiratory rate? Yeah, Shannon's got it. Your breathing is absolutely going to be affected, right? You're going to start breathing very heavily. Your rate is going to go up. You're going to take more breaths and you're going to attempt to take deeper breaths. You've heard it. You've probably done it. I know I've done it. I've been in situations when I'm exercising where I'm breathing because I'm not getting enough oxygen for the amount of work I'm doing. If you push it, if you keep going, even though you're breathing heavily, what's going to happen is there's going to come a point where you are not able to deliver the oxygen that you need in that moment. Your skeletal system particularly consumes an awful lot of oxygen, making an awful lot of ATP to keep you running. So if you cannot make enough ATP for yourself because your oxygen level, sorry about that, because your oxygen level is insufficient, you're going to start to ferment. Your cells are going to switch and they're going to start fermenting. Now, humans do a kind of fermentation called lactic acid fermentation. And I laugh because if you've ever been in that situation where you're running for exercise and you're breathing very heavily, but you're determined to keep going, one of the next things you're going to notice is that your legs are going to start to burn, right? Especially your quadriceps muscles, because they're one of the biggest muscles you have in your body. Your quadriceps muscles on the front surface of your thighs are going to start to burn. And the reason they burn like that is because the cells in there are fermenting and they're producing a molecule called lactic acid. And it's an acid, and it's going to irritate the tissue in your legs. Now, when you stop running, your breathing doesn't go back to normal right away, does it? When you stop exercising vigorously, especially if you're out of shape, you're going to continue like that for a few minutes, right? Until your breathing will start to slow and then it will return back. You've got to You've got a debt to pay to your cells. You're going to have to switch back from fermentation back to respiration as more and more oxygen becomes available to your body. But while you are exercising to the point of breathing heavy, you're actually going to move into fermentation because you simply cannot take in enough oxygen to do respiration. So you haven't stopped breathing. In fact, quite the opposite. You're breathing very heavily, but you can't get enough oxygen in that situation. And so you'll ferment. And the problem for you and I is that not only does it hurt while you're doing it, while the lactic acid builds up, but oftentimes we're also taxing our muscles beyond what they're able to do. And we're also sore for the next couple days because we put our skeletal muscles through things that they're not quite ready to do. Listen, I've been a weekend warrior myself, and I still do it from time to time. If it's a beautiful day outside, I may try to push myself to do a little bit more than I'm able to do. So human beings ferment. We don't ferment as our primary way of making ATP. We ferment when we cannot get enough oxygen. We cannot get enough oxygen to do cellular respiration. So fermentation for us is a secondary pathway. It's a second choice pathway for our cells, but it is there if we need it. Now, there are other organisms that will do fermentation similarly to you and I. Remember those bacteria and archaea that I mentioned? Now, they're not going to be over-exercising, but they may find themselves in an environment where they can't get their final electron acceptor. Maybe they can't get enough nitrate, or they can't get enough sulfate. They can switch over to fermentation as well. Depending on the species, they're going to do different types of fermentation. I said that you and I do a type called lactic acid fermentation, and there are some bacteria and archaea that do lactic acid fermentation. But there are lots of other fermentation pathways as well that you see in other organisms. One of the most interesting things about fermentation, I think, is that there are some microbes, bacteria and archaea again, that do fermentation exclusively. They don't ever do cellular respiration. They have evolved to just go to fermentation. That's what they use. And they survive quite well on it. You and I would not be able to survive on fermentation. It simply doesn't produce enough ATP. And that's what we'll talk about next. So if you cannot get your final electron acceptor in enough of a volume, whether that's oxygen or some other molecule that you use as your final electron acceptor, if you can't get it in enough of a volume, what's going to happen is the electrons that are brought in by NADH and FADH2, they're going to go down the electron transport chain and they're going to have nowhere to go. Remember, we said if oxidation occurs, reduction must accompany it. If there's no final molecule to get reduced, the electron transport chain will stop. And if the electron transport chain stops, that means there won't be any more electron acceptor molecules available because they're all going to be full of electrons. And that means there's not going to be any ATP produced by ATP synthase. And that is a life-threatening situation, not just for you and I, but for all organisms that use cellular respiration. Lucky for us, there is a secondary pathway called fermentation. Fermentation is simply a different metabolic pathway. That's all it is. It's a different metabolic pathway that's going to do two things for a cell. Most importantly, it's going to produce a little ATP. And the other thing it's going to do is it's going to regenerate our electron carrier molecules. In other words, it's going to give those electrons a place to go. NADH is going to have a place to donate its electrons so it can go back to being NAD+. Now, the ATP that's produced during fermentation is produced in glycolysis. And that's because glycolysis occurs in fermentation as well as cellular respiration. Glycolysis is going to keep happening during fermentation. And as we now know, a little bit of ATP gets made in glycolysis, a little bit of ATP. So fermentation does two things for a cell. It makes a little bit of ATP to keep the cell alive, and it regenerates electron carriers. I said that there are a lot of different fermentation pathways. You can go into microbiology textbooks, and there are charts and charts of different fermentation pathways that are performed by different species. But they all have a few things in common. Fermentation involves glucose being converted into pyruvate as the first part of the series of reactions. And this is just glycolysis. Those 10 steps are going to occur, breaking this 6-carbon molecule called glucose into 2 of these molecules called pyruvate. And along the way, a little ATP is going to get made. Remember, the electrons that get released from the glucose as it's broken down will be received by NAD+, and those will become NADH, all right, just like we saw in respiration. Now, here's where the pathway diverts. The pyruvate does not go through pyruvate processing. Instead, the pyruvate is going to be diverted into a different reaction, creating some different product. Remember, normally we expect pyruvate to become acetyl-CoA and a little CO2. That's what happens during pyruvate processing. During fermentation, the pyruvate is just going to be converted into something else. And along the way, the NADH will donate its electrons to make that product. So every fermentation pathway follows this basic route, if you will, this basic reaction series. Starts with glycolysis and then makes some kind of molecule that takes on electrons from NADH. Of all the different kinds of fermentation, there are 2 kinds that appear most often. So if you look at all the different organisms on our planet that are capable of fermentation, the 2 pathways that show up most often are lactic acid fermentation and another one called ethanol fermentation. And yes, this is the ethanol that we like to drink. So fermentation is occurring when pyruvate is going to get sent along a different pathway so that this molecule called NADH can get rid of its electrons somewhere other than the electron transport chain. NADH will be converted back to NAD+. That's going to be important when respiration begins again. And glycolysis is going to be making a little ATP for the cell. Remember, that process is called substrate level phosphorylation. It's not a very good way to make ATP. Fermentation is not nearly as efficient as cellular respiration is. Now, in lactic acid fermentation, and again, this is the one that you and I do, The pyruvate that gets made through glycolysis is going to accept electrons from NADH and become a molecule called lactate. That's the product. Remember from chemistry, lactate is another word for lactic acid. Whoops. So what's coming out of lactic acid fermentation is lactic acid, a little bit of ATP, and our electron carrier ready to take on more electrons. Ethanol fermentation is just a tiny bit more complex. There's one extra molecule involved in ethanol or alcohol fermentation, if you want to think of it that way. The pyruvate is first going to be turned into a molecule called acetaldehyde and a little bit of carbon dioxide. It's the acetaldehyde that is going to take the electrons from NADH, allowing it to become NAD plus again. And the product molecule, the acetaldehyde, is going to become ethanol. So there's sort of this middle molecule in here called acetaldehyde. Now, if you know anything about what happens inside the human body, acetaldehyde also gets made when ethanol gets broken back down. So when you and I drink ethanol, and that ethanol goes into our liver to be metabolized, that acetaldehyde molecule shows up again. It shows up as the ethanol is broken down, so it can be eliminated from your body. One of the interesting things about acetaldehyde is that it's very toxic to living cells. In fact, it's believed that acetaldehyde is one of the reasons that we feel so badly the day after we overindulge in alcohol. All of the alcohol that we drink, all of the ethanol that we drink has to be broken down because alcohol is a toxin. There's no use for it in the human body. And as it's broken down and it's making acetaldehyde, the acetaldehyde just causes inflammation in our body. So part of the reason you feel so lousy when you're hungover is because of this acetaldehyde molecule. So acetaldehyde shows up during alcohol production in fermentation. And it also shows up as ethanol is broken down when we consume it. So the last thing we'll do is we'll take a look at these two very popular kinds of fermentation. Lots of different organisms do these particular kinds of fermentation. This first slide, I know it looks very busy, but this first slide just diagrams for us the process of lactic acid fermentation. So the molecule glucose is going to go through glycolysis in order to make this molecule called pyruvate. That's nothing unusual. That's going to happen all the time in our cells, whether they're doing cellular respiration or fermentation. Remember, during glycolysis, we're going to make a little ATP through this process called substrate level phosphorylation. It's not a great way to make ATP, but the cell will take it because it's ATP and cells need ATP. The other thing that's going to happen during glycolysis is glucose is getting oxidized a little bit. So we need a place for the electrons to go. As we break apart bonds in glucose in order to make pyruvate, the electrons will be taken on by NAD+, and NAD+ will become NADH. We've loaded our electron carrier here. Normally, we would expect this NADH to donate those electrons into the electron transport chain. But we're going to imagine that for some reason, this person is not getting enough oxygen right now. And so the body's going to switch over from respiration to fermentation. So this pyruvate is not going to go through pyruvate processing right now. There's just not enough oxygen available to keep doing cellular respiration. So instead, pyruvate is going to get diverted along a fermentation pathway, and that's what this is showing us. It's going to get converted to lactate or lactic acid. Now, the pyruvate will accept electrons from NADH and allow it, that molecule, I keep clicking, sorry, allow that molecule to go back to NAD+. That's good. Cells need this for cellular respiration once it starts up again. And along the way, again, a little ATP gets made. So glucose goes in, lactic acid comes out, NAD comes out, NAD+ comes out, and a little ATP comes out. And that's lactic acid fermentation. Lots of animals do lactic acid fermentation, and some bacteria and archaea do it as well. It's quite a common pathway. Couple things to note about our weekend warrior. It's lactic acid that makes your skeletal muscles hurt while you're strenuously exercising. Now, the cell is going to get rid of that lactic acid. It's going to get metabolized and essentially sent backwards through the fermentation pathway. It will be converted back to pyruvate. And then at some point, once there's enough oxygen around, that pyruvate will go into pyruvate processing and down the pathway of cellular respiration. It can take about 24 hours, depending on how strenuous the exercise is. It can take about 24 hours for your body to get rid of all this lactic acid that builds up. So that pain that you feel during the exercise, that's definitely from lactic acid. But the pain you feel the next day, and even for the next couple days after strenuous exercise, that's not from lactic acid. That's actually from overusing your skeletal muscles. If you overextend or overflex your skeletal muscles beyond their capacity, beyond your level of fitness, you're actually going to cause some microscopic tearing inside your muscles. And I don't know if you've had any anatomy in your education yet, but there's a structure inside your muscle called sarcomeres. And it's the sarcomeres that actually contract and cause your muscles to shorten. Those microscopic tears are happening to these sarcomeres. And when they get torn from strenuous exercise, they're going to get inflamed. And inflammation causes pain. It's going to take a couple days for those micro tears to resolve. But interestingly, if you keep going with your exercise, your muscles will get stronger and stronger, right? You'll become more fit with more exercise. You'll build more sarcomeres inside your skeletal muscles so that the next time you go out for your 10-mile run, hopefully you won't be nearly as sore as you were that first time. And the more you stay with it, the more fit you get, it'll be easier and easier to do this exercise routine. And you will be less likely to start fermenting while you exercise, the more fit you become. And that's good news, because then you don't have to deal with the soreness that comes along with it. Lactic acid that builds up during lactic acid fermentation, it's just going to get broken down. It's going to get broken down by your cells and gotten rid of by essentially sending it backwards in that pathway and converting it back to pyruvate. So if you look at this diagram, over here on the right, this represents what's happening in muscle. Over here, this represents what's happening in the liver. And then we've got the bloodstream sort of in between. So glucose gets delivered to your muscle cells during vigorous exercise, and that glucose gets sent through glycolysis to make pyruvate. If you're not getting enough oxygen in that moment, then you're going to ferment. That pyruvate is going to get converted to lactate or lactic acid. That lactic acid will go into your bloodstream. It's going to make your muscles hurt in the short term while you're exercising, but within about 24 hours, it will be transported to your liver, and it will be sent through a metabolic reaction that converts it back to pyruvate. And interestingly enough, pyruvate can actually then go into several different metabolic reactions. It just depends on what needs your body has at that moment. But one of the things that can happen to pyruvate is it can be converted back to glucose. This is a process called gluconeogenesis. It's a long word, but if you translate it, it just means carbohydrate, new, and creation. So we're building new glucose through this reaction. Now, you're not going to build glucose unless you need glucose. So most of the time, our liver doesn't do gluconeogenesis with pyruvate. If you have plenty of glucose circulating in your blood already, the liver cells are going to do something different with the pyruvate. But it is possible to take lactic acid and turn it right around and make glucose again. I put these pictures in just to remind us that a lack of oxygen or not having enough oxygen doesn't only happen to weekend warrior athletes. There are a lot of athletes who are at the top of their game physically, right? They are as fit can be. And yet they have to deal with lactic acid fermentation too, because they are performing at a level where they're simply not able to get enough oxygen into their cells to make enough ATP for the work that they're doing. A really good example of this is short distance running, sprinting. Olympic level sprinters are going to ask their skeletal muscles to perform at a level that they simply cannot do while making ATP on the fly, while making ATP on the go. So these folks are going to ferment. Even though they are at the top of their game, they are going to do lactic acid fermentation. One of the things you'll notice at high performing levels is that after an athlete performs a race, they'll often have an athletic trainer massaging their muscles. They're big muscle beds. So think about the quadriceps muscle again in the thighs and the muscles in the lower back, up in the shoulders. What this person is trying to do is speed along the process of getting that lactic acid out of the muscle cells into the blood so it can go to the liver and get converted back to pyruvate. Very interesting stuff. Now, I also wanted to point out, just because it's, I think, interesting, there are other reasons that lactic acid can build up besides vigorous exercise. There are disease states where the body doesn't get enough oxygen to do cellular respiration. We talked about the person who falls down from a heart attack, but there are also a lot of disease states where the person is just so sick that they're not getting enough oxygen into their tissue. The term we use for that is poorly perfused. There's just not enough oxygen getting delivered to the tissues in the body during certain disease states. That person is also going to do lactic acid fermentation, even though they're certainly not exercising in that moment. Certain kinds of traumatic injuries, massive blood loss, certain kinds of uncontrolled diabetes, people who have uncontrolled high blood pressure or hypertension. These people are prone to not having enough oxygen for cellular respiration because of poor tissue perfusion. So they tend to do fermentation more than a healthy person would. And you're going to see that in their blood. Physicians will draw blood from folks like this, and what they'll see is a high level of lactic acid in the blood. And that tells them that they are not metabolizing normally in that moment. Something's wrong. And since they're not exercising, it must be a disease process that's causing this problem, not getting enough oxygen to their tissue. Now, the other kind of common fermentation I mentioned was ethanol. And that's what this diagram shows us. Remember, there's just an extra step in ethanol fermentation that we should be familiar with. Glycolysis is still going to occur. So glucose is going to be sent through the reactions of glycolysis to create pyruvate, just like it would during normal cellular respiration. Along the way, a little ATP gets made, and NAD becomes NADH as it accepts electrons from glucose. Now, the pyruvate is not going to be directly converted to ethanol, but first it's going to be converted into acetaldehyde and a little carbon dioxide. It's the acetaldehyde molecule that will then accept electrons from NADH. That's what this is showing us. And it gets converted into ethanol. Acetaldehyde is considered a toxin to our human cells. So is ethanol. Both of those molecules are considered toxic to human cells. Fermentation is a lousy way to make ATP because the ATP is made using substrate level phosphorylation instead of oxidative phosphorylation. It's just not as good. Fermentation is going to make 2 ATP per mole of glucose compared to, remember, somewhere between 30 and 36 ATP per mole of glucose in respiration. Sorry, I keep doing that. So cellular respiration and oxidative phosphorylation is the best way to make ATP in our human cells because it's a very efficient mechanism. When we can't do it, though, we have a secondary pathway that we can switch to temporarily, and that's fermentation. Most organisms, including you and I, won't use fermentation if we can use cellular respiration. Cellular respiration is a better pathway for us, so we will preferably use that. As long as there's oxygen around, we're going to do cellular respiration and oxidative phosphorylation. But we have fermentation there just in case. There are lots of microbes like us who can use both respiration and fermentation, preferring respiration. There are other microbes that consider respiration and fermentation to be equally good. And there are still other microbes that only ferment. That's the only pathway they use. Very interesting. So if you've ever heard of a microbe called a facultative kind of organism, that's a microbe that can switch back and forth between cellular respiration and fermentation equally well. E. coli, for example, down in your intestine. E. coli can just switch back and forth. It can do respiration when times are good for respiration. It can do fermentation when times are good for fermentation, and it just switches. And it's happy to do either pathway. You and I are different. Our cells are going to do respiration if it's at all possible. We only use fermentation as a backup plan. All right. So that's all the information I have for you today. When you have time to review it, If you have any questions that came up today, just jot them down and we can certainly talk about them when we get together on Tuesday next week. Or you can always message me and send me questions as they arise. Remember, we're not meeting on Thursday for lecture because it's Veterans Day, but I will see you on Tuesday next week. And remember that a week from Thursday is your next lecture exam. Okay? Any questions at all? You're welcome. Any questions at all before I let you go?
Thank you so much.
You're very welcome. Thanks for coming, you guys. I appreciate it.
Bye. Have a nice day.
Have a good evening. We'll see you next.
Thank you. Have a good one.
Thank you. Have a good one.